Hello, and welcome to my micro talk for my poster for this Molecular Life of Diatoms conference. I'll be presenting my work so far on my PhD project, where we're using transcriptomics to elucidate the survival strategies of century-old diatoms in sediment. The diatom we're looking at, Skeletonema marinoi, is a chain-forming marine-centric species found commonly in temperate coastal waters. One of the reasons we're using this species as a model is that its resting stages can be revived from sediment cores after decades or longer, so we can compare historic and contemporary samples. However, we still don't know exactly how they're able to survive in dark anoxic conditions for so long, which is what we're hoping to figure out in this study. To that end, we've performed a differential expression analysis to compare gene expression in vegetative and resting cells, looking for genes and pathways relevant to resting stage survival. 2 axenic strains of S. marinoi were put into dark anoxic conditions without nitrate for 49 days to induce resting stages, at which point nitrate and bicarbonate were added. After this, the dark anoxic conditions were maintained up to a total of 189 days. During this process, RNA was sampled at 7 time points and Illumina libraries were prepared and sequenced. We performed differential expression analysis based on the current version of the Skeletonema marinoi R05AC reference genome annotation, which contains 17,383 gene models. In terms of upregulated genes, 816 of these 17,000 were upregulated at all resting stage time points in both strains, of which 559 have some kind of functional annotation. Performing Go enrichment analysis, the most significantly enriched Go terms were related to ribosome biogenesis and non-coding RNA metabolism, as well as metabolic processes of various compounds, possibly storage compounds. One gene which appeared in the top 10 most upregulated in all comparisons encodes a fatty acid desaturase, which is consistent with observations in other diatom species resting stages where increased amounts of unsaturated fatty acids were found. Comparing the 49-day time point with those after nutrient addition, the main upregulated terms pertain to fatty acids and amino acids. One gene was upregulated at all time points in both strains after nutrient addition, albeit this gene's function is currently unknown. Looking at the downregulated genes, we found that 1,437 of the 17,000 gene models were downregulated at all time points in both strains, of which around half had some level of annotation. The most significantly downregulated Go terms among these 1,400 were related to the cell cycle and DNA replication, which stands to reason if the cells aren't dividing. And although it's not shown on the figure here, photosynthesis-related processes were also significantly downregulated, which also makes sense. While no genes were consistently among the top 10 most downregulated genes in all time points across both strains, when looking at those downregulated after addition of nutrients, both a fucoxanthin, chlorophyll AC protein, and a protein of unknown function are among the top 10 in all comparisons. So, a few of my takeaways at this point in the project. Our observation that genes involved in fatty acid metabolism are upregulated in the S. marinoi resting stages appears to be consistent with the changing fatty acid composition of resting stages observed in other diatom species. Somewhat less consistent is the fact that ribosome biogenesis appears to be so significantly upregulated in resting stages, because looking through the literature, most diatoms seem to downregulate this process in response to nitrogen starvation. I only found one other example in the literature of ribosome biogenesis upregulation in resting stages, in Cattosaurus socialis, although the study in question was performed across a shorter timescale than ours. So the question is, why is this process upregulated when the cells are supposed to be going dormant? There are a few possibilities here that I'd like to investigate. Are the ribosomes being used as an additional store of nitrogen in addition to the nitrate found in the vacuole? Are the ribosomes being produced and then put into a hibernation state, so that once the resting stages are revived, they can quickly resume vegetative growth? Or is this observation just a result of many differentially expressed genes lacking a functional annotation? With that in mind, I'm currently in the process of manually curating the S. marinoi gene models and applying other annotation tools to hopefully obtain more information about these genes relevant to resting stages. In particular, I'm hoping to identify additional genes relevant to S. marinoi's nitrogen metabolism as part of the wider Survivors of the Sea project. Thank you very much for listening, and please let me know if you have any thoughts on these results.